Chapter 1, the Biotechnology Century and its Workforce. This video is going to cover all the vocabulary and definitions that are seen in this chapter. We're not going to be reading anything else but the blue bolded vocabulary terms. And this first one is genetically modified foods, or GM, which are foods derived from sources that have been genetically modified and must be approved for consumption. Next, we're going to scroll through the entire chapter. Animal biotechnology. Animal biotechnology, a diverse discipline of biotechnology that involves the use of animals to make valuable products such as recombinant proteins and organs for human transplantation. Also includes organism cloning. Next, we got section 1.1, what is biotechnology and what does it mean to you? We have the term biotechnology. A broad area of science involving many different disciplines designed to use living organisms or their products to perform valuable industrial or manufacturing processes or applications that will solve problems. Okay, and fermentation will be next. A metabolic process that produces small amounts of ATP from glucose in the absence of oxygen and also creates byproducts such as ethyl alcohol, known as ethanol, or lactic acid, known as lactate. Fermenting microbes, bacteria and yeast, are important for producing a variety of beverages and foods, including beer, wine, breads, yogurts, and cheeses. Next, we look at selective breeding. Selective breeding, mating organisms with desired features to produce offspring with the same characteristics. Next is model organism. Model organisms are non-human organisms that scientists use to study biologic processes in experimental laboratory conditions. Common examples include mice, rats, fruit flies, worms, and bacteria. And the next term that we look at is in vivo. In vivo means occurring within a living organism, from Latin, in something alive. And then we have antibiotic. Antibiotic, a substance produced by microorganisms that inhibit the growth of other microorganisms, commonly used to treat bacterial infections in humans, pets, and farm animals. Now we have batch processes or large-scale processes, or scale-up. That's an important alternative word for this word, scale-up. Growing microorganisms such as bacteria or yeast and other living cells such as mammalian cells in large quantities for the purpose of isolating useful products in a batch. Next is gene cloning. Gene cloning, the process of producing multiple copies of a gene. And genetic engineering, the process of altering an organism's DNA. This is usually by design. Human Genome Project, sometimes referred to as, in capitals, acronym HGP, an international effort with overall scientific goals of identifying all human genes and determining their locations to each human chromosome. And when they determine their location, they refer to it as mapping. So some scientists will say It's an effort with overall scientific goals of identifying all human genes and mapping their locations to each chromosome. Next, genome. Genome is all of the genes in an organism's DNA. Here we got a good shot of figure 1.2 gene maps of chromosomes 13 and 21. Okay, the next term defined: basic sciences. Research disciplines that examine fundamental aspects of biological processes, often without obvious direct applications, for curing disease or making a product. This term here, bioinformatics. Interdisciplinary science that involves developing and applying information technology, such as computer hardware and software, for analyzing biological data such as DNA and protein sequences. Bioinformatics also includes the use of computers for the analysis of molecular structures and creating databases for storing and sharing biological data. Alright, the next term is a company called Genentech. California company, name derived from genetic engineering technology. Founded in 1976 and widely recognized as the world's first biotechnology company. Okay, this next term, recombinant proteins. Recombinant proteins are commercially valuable proteins created by recombinant DNA technology and gene cloning techniques. Examples include insulin and growth hormone. Okay, the next term is cell culture. Growing cells in laboratory conditions outside of a whole organism. In vitro. Usually a term applied to growing mammalian cells. Next, fermenters. Fermenters, or bioreactors, are containers for growing cultures of microorganisms or mammalian cells in a batch process. Fermenting vessels allow scientists to control and monitor growth conditions such as temperature, pH, nutrient concentration, and cell density. Fermenters do not require oxygen when anaerobic organisms are fermenting nutrients. Bioreactors use non-fermenting organisms that require oxygen. And we have that same term, bioreactors. Bioreactors are cell systems that produce biologic molecules, which may include fermenters. Next. Gene therapy. Okay, gene therapy is the use of therapeutic genes to treat or cure a disease process. Also refers to the delivery of genes to improve a person's health. Okay, molecular farming. Molecular farming is the use of plants as sources of pharmaceutical products. Next term should be antibodies. Antibodies. Proteins produced in response to a non-self molecule by the immune system. Antigen-binding immunoglobulins produced by B cells that function as the effector in an immune response. Transgenic animals. Animals that contain genes from another source. For instance, human genes for clotting proteins can be introduced into cows for the production of these proteins in their milk. Okay, we got forensic biotechnology. The analysis and application of biologic evidence such as DNA and protein sequence data to help solve or recreate crimes. And DNA fingerprinting. DNA fingerprinting, an analysis of an organism's unique DNA composition as a characteristic marker or fingerprint for identification purposes, such as forensic analysis, remains identification, and paternity. DNA fingerprinting is also used in biologic research, for example, to compare related species based on their DNA sequences. Okay, next we have bioprospecting. Bioprospecting endeavors to capitalize on indigenous knowledge of natural resources. However, bioprospecting may also describe the search for previously unknown compounds in organisms that have never been used in traditional medicine. Medical biotechnology. Medical biotechnology is a diverse discipline of biotechnology dedicated to improving human health. It includes a spectrum of topics in human medicine, from disease diagnosis to drug discovery, disease treatment, and tissue engineering. Let's see. Gene therapy. Gene therapy, the use of therapeutic genes to treat or cure a disease process, also refers to the delivery of genes to improve a person's health. Here we have quality assurance, or QA. All activities involved in regulating the final quality of a product, including quality control measures. And then quality control, QC. Procedures that are part of the QA process involving laboratory testing and monitoring of production processes to ensure consistent product standards of purity, performance, and the like. Proteome. Proteome, the entire complement of proteins in an organism. Now we have mutations, a change in the DNA structure or sequence of a gene. Next is BRCA1 or BRCA1, a gene found in 65% of human breast tumors, commonly part of the breast cancer diagnosis for treatment, or it goes by BRCA1. DNA microarray. A chip consisting of a glass microscope slide containing thousands of pieces of single-stranded DNA molecules attached to specific spots on the slide. Each spot of DNA is a unique sequence. Gene chip. Okay, so it says it's synonymous with DNA microarray. Pharmacogenomics. Pharmacogenomics is a form of customized medicine in which disease treatment strategies are designed based on a person's genetic information for a particular health condition. And chemotherapy. The treatment of cancer and other diseases with specific chemical agents or drugs that have a toxic effect on diseased cells or disease-causing microorganisms. Metabolomics. A biochemical snapshot of the small molecules produced during cellular metabolism, such as glucose, cholesterol, ATP, and signaling molecules that result from a cellular change. Embryonic stem cells, ESC. Okay. Yeah, embryonic stem cells, ES cells, are collected from the inner cell mass of blastocysts. Transformed ES cells are usually injected into the inner cell mass of a host blastocyst. Okay. Induced pluripotent stem cells, iPSCs. Nuclear reprogramming of mouse and human cells, heralded as a revolution in stem cell biology research. One approach has involved using retroviruses to deliver 4 transgenes, OCT34, SOX2, C-MyC, and KLF4 into fibroblasts. Expression of these 4 genes, which encode transcription factors involved in cell development, reprograms the fibroblast back to an earlier stage of differentiation. iPS cells demonstrate many properties of HESCs, such as self-renewal and pluripotency, and appear to be indistinguishable from HESCs. Regenerative medicine, a discipline of medical biotechnology that involves repairing or replacing damaged tissues and organs by using tissues and organs grown through biotechnological approaches. Research and development, R&D. All of the processes involved in basic research, for example, preclinical research and development of a potent product or potential product. The lifeblood of a biotechnology company, research and development, is how companies identify new technologies, drugs, etc. for commercialization. Pharmaceutical companies. Companies creating drugs for the treatment of human health conditions. Startup company. Formed by a small team of scientists who believe they may have a promising product to make such a recombinant protein to treat disease. The team must typically then seek investors to fund their company so they can buy or rent laboratory facilities, buy equipment and supplies, and continue the research and development necessary to make their product. Venture capital, VC. Funding for an enterprise provided by financiers who see promise in their enterprise and expect a substantial financial benefit for the high risk, or expect to take a loss. Angel investors. Investors that save your company from financial ruin at the last minute, like an angel. Initial public offering, IPO. First sale of stock by a formerly private company. It can be used by either small or large companies to raise expansion capital and become publicly traded enterprises. Okay, now we're getting to laboratory technicians. Laboratory technicians. Entry-level laboratory jobs with a range of responsibilities such as preparing solutions and mediums, ordering laboratory supplies, cleaning and maintaining equipment, may sometimes involve bench research. Research assistants. Research assistants are laboratory positions in which individuals are primarily involved in carrying out experiments under the supervision of other scientists such as principal or senior scientists. Research associates. Laboratory positions in which individuals are primarily involved in carrying out experiments under the supervision or of other scientists such as principal or senior scientists. Principal. Principal, or senior scientist, science leadership position in biotechnology companies. Senior scientists are usually PhD or MD, trained individuals who plan and direct the research priorities of a company. The principal scientist. Bioinformaticists, scientists specializing in the bioinformatics. Scale-up processes. The industrial implementation of processes in which chemical or microbiological conversion of material takes place that behave differently on a small scale, in laboratories or pilot plants, and on a large scale in production. Alright. U.S. FDA, Environmental Protection Agency, EPA. Or actually, no, the first one that we're going to do, it's a totally separate. No, it isn't. That's weird. FDA, I guess they're combining FDA and EPA together. The agency whose primary purpose is to protect human health and to safeguard the natural environment, air, water, and land, by working with other federal agencies in the United States and state and local governments to develop and enforce regulations under existing environmental laws. U.S. Department of Agriculture, USDA. Agency created in 1862 that has many functions related to the advancement and regulation of agriculture. Some of those functions include the regulation of pests, plants, and veterinarian biologics. Generic drugs. Generic drugs are copies of brand name products that generally have the same effectiveness, safety, and quality, but are produced at a cheaper cost to the consumer than the brand name drugs. Biosimilar drug. Biosimilar drugs are also called follow-on biologics, subsequent versions of a recombinant protein product after the original patent has expired. The biosimilar is produced by a different company than the innovator holding the initial patent. As a result, when a biosimilar is made, the exact production processes are not the same as the innovator's, hence the product or its manufacturing is similar, but not identical to the original protein. The equivalent of a generic of a pharmaceutical drug. Next we have customer relations specialists. Sometimes work in QA or quality assurance divisions of a company. One function of a customer relations is to investigate consumer complaints about a problem with a product and follow up with the consumer to provide an appropriate response or solution to the problem encountered. Product complaint specialists are quality control specialists that certifies that a product meets the criteria specified in its package insert or other specifications submitted to regulatory agencies. Next we have sales representative. Salespersons in biotechnology companies. Sales representatives are people persons who work closely with medical doctors, hospitals, and healthcare providers to promote a company's products. Marketing specialists. Marketing and sales position in biotechnology companies. Marketing specialists are often involved in designing ad campaigns and promotional materials to market effectively a company's products. Finance divisions. Unit responsible for overseeing the company finances. Legal specialists. In biotechnology companies typically work on legal issues associated with product development and marketing, such as copyrights, naming products, and obtaining patents. Staff in this area also address legal circumstances that may arise if problems are found with a product or litigation from a user or product. Wow. Okay. So that actually completes the vocabulary and definitions for chapter 1. Let's see some of these questions if we can answer these. Number 3, which area of biotechnology involves using living organisms to clean up the environment? I think that's bioremediation. Describe how pharmacogenomics will influence the treatment of human diseases. Pharmacogenomics involves prescribing a treatment strategy based on the genetic profile in a patient, fitting the drug to the patient's genetics. A RNA or DNA sample from a patient with a medical condition can be analyzed to determine whether the genes that person expresses match the genetic profile of a particular disease process. If they do, a specific treatment approach can be designed according to the best known treatment strategy based on the patient's genetic profile. Okay, in question 5, let's see. Distinguish between QC and QA and explain why both are important for biotechnology companies. No product can leave a biotechnology company until it passes rigorous tests by the internal unit called quality control. Quality assurance is responsible for monitoring everything that enters and leaves a company to ensure product safety and quality, and tracing sources of problems identified by complaints as specified by regulatory agencies such as the U.S. Food and Drug Administration, or FDA. QA and QC measures are important for every step of research and development, R&D, as well as for product manufacturing, including packaging and marketing. Let's see. 11. Explain how biotechnology companies got started. Include in your answer a description of how a startup company is funded. Alright, biotechnology companies often begin with a discovery by a scientist working in academia. For example, a gene or protein is found to be important in a disease process and may have potential applications as a recombinant DNA product. Starting a biotechnology company, a startup company, to develop this protein as a product then requires significant initial funding to do the R&D, such as preclinical research in humans and then ultimately FDA clinical trials, if the protein is to be used in animals. Sometimes a company starts with initial seed money, which may come from a few individual investors. Then, if the product shows potential, the person or persons starting the company will seek venture capital from corporations or groups wishing to provide money for early-stage, high-risk, potentially high-reward earning companies, or angel investors, the individuals who provide capital for a startup in exchange for ownership and provide making in the company. And 12. Describe the differences and similarities between the pharmaceutical industry and the biotechnology industry. So, pharmaceutical industry is primarily involved in producing, selling, and marketing drugs for treating humans. This is one similarity to many medical biotechnology companies. However, the way in which pharma is opposed to biotech makes such drugs differ. Pharma companies primarily use synthetic chemical processes to make drug compounds, whereas biotech companies use living cells to produce a drug. Pharma companies also often produce medical devices. In recent years, many large pharma companies have acquired smaller biotech companies. As a result, the many traditional pharma companies are also doing biotechnology R&D. For example, the pharma company Roche purchased the biotech company Genentech.